Welcome everyone to the South Kohala Coastal Partnership Speaker Series. And I'm Lauren Kupita, the coordinator of the partnership. And the goal of this series is to provide an opportunity to learn more about the partners and the work being done in South Kohala. And the catalyst that started this was the State Division of Aquatic Resources Coral Reef Working Group identified South Kohala as a priority area for the coral reef strategy to implement action to reduce anthropogenic impacts to the reef. And in 2018, over 30 organizations and agencies participated in creating the South Kohala Conservation Action Plan 2030 that outlines 6 specific strategies to guide the partnership's collective work towards restoring and protecting coastal resources. Today, we are excited to have Dr. Shawn Larson, Senior Conservation Research Manager with the Conservation Programs and Partnerships at the Seattle Aquarium. And Shawn launched the Seattle Aquarium Conservation Research Program in 1995 and now includes over 20 long-term research projects, and her research spans a variety of areas in conservation research projects in the Salish Seas and in Hawaii. Shawn's research is cross-disciplinary and collaborative, including projects focused on marine mammal physiology, ecology, and genetics, sea otter distribution, population abundance, and foraging patterns, shark and octopus conservation, physiology and genetics, and temperate and tropical reef fish population trends and benthic ecology, and orca population trends and conservations. And Shawn will be presenting data from a multi-prong study on coral reef ecosystems in West Hawaiʻi.
Thank you, Lauren. That was a great introduction. I'm going to share my screen and start my presentation. Let's see, everything looks good?
Yes.
Okay, perfect. Okay, yeah, as Lauren said, I would like to share with you a multi-pronged study of the coral reef ecosystems in West Hawaiʻi that I started in 2009 and have had help from many staff members and community partners, staff members at the aquarium, as well as community partners in Hawaii over the years. And part of this research was part of one of my technician's master's thesis, Amy Olson. So let's see if I can get this going. Okay, so what is the conservation programs and partnerships at the Seattle Aquarium? Well, this is a relatively new department at the Seattle Aquarium. It was created 5 years ago. I used to work in the husbandry department. But we have a new director, Dr. Erin Meyer, and she oversees 3 senior managers. I am the research manager. We also have a senior ocean policy manager who has a technician, and they go to both state and federal legislative sessions and listen to bills and decide which one the Seattle Aquarium should support to further ocean conservation. We also have a species recovery manager who does work in the Salish Sea with abalone and sea otter recovery, as well as zebra shark recovery and augmentation in Indonesia. And so I have a small but growing research department, and these are the folks that work under me. I just hired a new clean seas research scientist who studies marine pollution. And I have a climate resilience research scientist who studies climate change. And then Amy Olson is my research technician who helps me move all of my 20+ conservation research projects forward. And we also have research support divers. And we're constantly adding new staff members to this growing team. We recently added a research technician to assist Zach Randell with his climate change research, and have added another research diver and a temporary research technician. So it's a growing department at the Seattle Aquarium, and this department does not do any work with animal husbandry or exhibits. It's purely focused on conservation programs. We have a bunch of volunteers that assist us as well. So all of our conservation research has to fit within our 3 conservation priorities, and those are climate resilience or climate change, sustainable seas or overharvest, and clean seas or marine pollution. So why are we doing research and monitoring in Hawaii? Well, the Seattle Aquarium has exhibited Hawaii reef fish and corals for over 40 years in our Pacific Coral Reef exhibits. And we also, we inspire and educate our guests about the animals and the ecosystems in Hawaii. We also sponsor a Hawaii weekend with one of our partners, Alaska Airlines, and this is in the winter when people in Seattle really want to get away to a warmer climate. And we talk about the importance of healthy Hawaii reef systems and healthy oceans around Hawaii, and then all the animals that inhabit it. And we basically, in 2009, I decided that I wanted to do more for Hawaii conservation. So we started a monitoring program in 2009. And this is our 15th year of conducting surveys in West Hawaii. And we've had many staff members participate in this. These are all the staff members that have participated in this 1 week survey that we do annually in February. We also have partners on the ground in Hawaii with Division of Aquatic Resources. We initially started doing this work with Dr. Bill Walsh with DAR, and more recently, Chris Teague. We've been working with Tracy Wigner at the University of Hawaii Hilo for about 10 years now, sharing our fish data and coral data, and trying to merge it with her water quality data to get a better understanding of what's happening in these reefs. And then we've also been working closely with Brian Tissot, who initially started the West Hawaiʻi assessment surveys with Bill Walsh back in the 1990s. And then we have other community partners that assist us with boat access and housing and diving. So it really does take a village. So what is our project about? We basically have 3 parts of this project. One is we use a unique survey method that no one that we know of is using in West Hawaii. The second is to analyze fish community change over time, specifically after the marine heat wave events, the marine heat waves that occurred in 2014 and 15. And we also wanted to analyze coral community changes after this marine heat wave events. So the management area in West Hawaii is complicated. There are areas that are essentially marine protected areas and closed to all extractive activities like fishing and aquarium collecting. And then there's other areas, the blue is the fisheries replenishment areas that are semi-closed to any extractive activities. And then there are areas that are fully open to fishing and etc. And these are the 8 sites that we've been surveying since 2009. And our initial question was, how do the different management schemes in each of these areas affect fish populations? And so when we first started this work, sites 6 and 7 up here in Mahukona were mostly open areas to fishing and collecting. The Puako sites here in the middle were fish replenishment areas or semi-closed. And then sites 3 and 4 are off of Old Kona Airport. And this was a marine life conservation district, mostly closed to fishing. And since we started this work, most aquarium collecting activities have been stopped, although there are some permits available to collect. They're very difficult to get. So what is our method? Well, we use a 100-meter diver operated video. And this was one of our first years. We swim 1 meter off of a vertical reef and 1 meter off the bottom. We have a camera operator. So we're taking pictures, video of the reef fish communities and the coral when we're doing this. And we have then the camera operators closest to a vertical reef, and that's we just follow that vertical reef as our route. And then to the outside of that camera operator is a reel operator, and that reel diver starts in the middle of our survey area, which is marked by GPS coordinates and unique features on the bottom. We don't leave anything on the bottom to permanently mark this site, but we're able to identify that start position within about a meter to start these surveys. We swim 50 meters one direction, stop, and then swim 50 meters back to the beginning, videotaping the whole time. And in this way, we can capture fish behavioral changes with before a diver swims through and then after a diver has swum through to see if we are pushing fish out of the area. And then we go another 50 meters the other direction and then come back. We do this, and we do that the 50 meters instead of a full 100-meter transect is because this transect tape is about an inch wide, but just deploying 100 meters of even an inch wide transect tape underwater, it does create some resistance, and especially if there's surge can actually be quite hard to handle. So 50 meters is a much easier length of transect tape to handle. We do this once per year at the same time of year. And another thing that we were doing is we were doing 4 replicate transects per site each year to try and gather those schooling fishes that are coming and going, and also that rare fish that is popping up. And then since we have video, we're not trying to count the fish with a slate that day. We're taking that video back to the aquarium, we're putting it up on a large screen, we're slowing down the videotape, and we can zoom in, etc. And that way we can try and capture and count as many of the different fish species and their relative abundance as possible. Again, we're wearing full face masks, so you can see here the diver wearing a full face mask. This way we can talk to each other, and it acts as an audio slate, essentially an audio stamp on that video. So if there is a very rare fish or a fish that's in the shadow, the diver that day can suggest what that species is, and then the fish counters back at the aquarium can determine whether or not they can actually count that fish off the video. So here is a little, hopefully this will work, video of our method, and I'm trying to get rid of my pointer here. It's not letting me. There we go. Okay, so this is what it looks like. You can see we've got the video diver swimming next to the reef face, about a meter off the bottom and a meter off of the reef. In this way, in the field of view of the camera, we can see the fish associated with the bottom, the benthos. We can see the fish associated with the reef, vertical reef, and then we can see fish associated with the open water. Okay, so what did we find in our methods? Is one transect enough? What I wanted to point out here is that for Pu'uko, Okona, and Mahukona over time, the total number of individual fish that we've counted off these surveys has increased over time, with a peak here in Olakona about 2014-15, as well as the species richness. And here on the bottom, we got into some modeling about like how many transects we really needed to capture all of the most fish community structure. And you can see here, this is where the curve starts to flatten, and it's about 10 transects. And we just don't have enough time when we're there once per year to do 10 transects. So we're thinking, well, maybe, the 4 we're doing is enough. But then when we looked into multivariate space, and basically each one of these dots is 1 transect each year. And you can see in multivariate space, Mahukona takes up this much space, Pu'uko takes up this much space, and Old Kona takes up less space. What you can also see is Pu'uko and Mahukona are overlapping in this multivariate space a bit. And what this just shows you is that the dots that are closer together have similar community structure. So we're like, okay, we can see that there's some community structure here that these 4 transects each year is informing. But if we just looked at it with one transect, you see the same patterns generally. So we're like, okay, 4 transects isn't enough, is not needed, although they do capture more community structure. It's not necessary to document changes in these fish communities. So that's great. Once we did that, we'd figure, okay, now we have more capacity to add other sites. And this is just another way of showing you that. So these are all the different sites. And these on the left is the multivariate space with 4 transects, and this is a multivariate space with 1 transect, and you can see the shapes are very similar. And what I want to point out here is that Old Kona takes up less space than Pu'uko sites as well as Mahukona, and that just means it's more stable over time. So, well, let's dig in a little bit more into the fish. So that was just kind of a methods assessment into this unique methodology. What about the fish? So we investigated changes in relative abundance of reef fish before, during, and after a marine heat wave, and that was the marine heat wave in 2014-15. And we explored how these changes in functional groups influence their fish assemblages. And this is just a graphic to show that ecosystems can change over time, and they can change from one state. This is a healthy state where the ecosystem is in this pocket, but if it gets disturbed enough by, say, like climate change or changes in ecosystem structure like coral bleaching, this healthy state can be bounced down to a different state, which may eventually be less healthy. So with climate change and increases in sea surface temperature, we can get coral bleaching, which can change the coral reef habitat, eventually changing the ecosystem services, which may be fish for people to eat, and also tourism. And then this changes the ecosystem values. So this is why we like to see how ecosystems respond to these climate change events. So my tech Amy Olson did this for her master's degree, and she looked at these different functional groups in these areas in Hawaii that we have been monitoring. So we have predators, secondary consumers, planktivores, those that eat plankton, coralivores, those that eat coral, browsers, grazers, and scrapers, and those are all that feed on algae. So how were these fish assemblages impacted by the marine heat wave? And again, this is another multivariate graph that shows we've got Mahukona over here in the solid green. We've got Pu'uko down here in the other lighter green, and then we've got Kona here, and these solid circles are pre-marine heat wave state, and you can see that they are spread out a bit, but then post the marine heat wave, everything becomes more similar. But what I want to also note, so Mahukona and Pu'uko are becoming more similar to Kona, but Old Kona really didn't change that much. So Old Kona seems to be more resilient to climate change events. And then when we looked at abundance by area, here's Mahukona, here's Pu'uko, here's Kona, and this line and the red asterisk is the marine heat wave event. And what we want to note is that generally, which is kind of surprising, is the fish numbers are increasing after the marine heat wave, except for Old Kona has a lot of interannual variability and is relatively stable. And then this is just looking at some of the top 3 functional groups by area. And you can see the grazers, the planktivores, and the secondary consumers or detritus eaters. By far, this fish functional group that increased the most were the grazers over all three areas, and that's probably because of increased algae. Whereas Kona here, there was the planktivores have been pretty plentiful and actually pretty stable over time, as well as secondary consumers. Whereas here at Mahukona, the detritus eaters or secondary consumers increased, as well as at Puako. So, but what happened to the coral? So the story of the fish abundance post-marine heat wave seems pretty positive. But what happened to the coral? And again, we use these diver operated video to look at the benthos as well. And how has the coral cover diversity and health changed over time on these reefs? And then this slide just gets into the coral bleaching. A healthy coral has symbiotic algae in it, zoanthillae. I think I'm saying that right. That then in a stressed coral, for some reason, they spit out those symbiotic algae, and it's that algae that actually gives the coral some of its color and also some of its nutrients. So when a coral becomes bleached, it's not necessarily dead, it's stressed and it can recover. But if the coral is left bleached and vulnerable for a longer period of time, then it does eventually die. So this is a Cropera coral at one of our sites, site 8. You can see here prior to the marine heat wave, we've got a healthy Cropera plate coral. 2016, part of it is bleached, and then 2018, this is the same coral and it is dead. And then this is an area in Puako. 2009, we've got the lobe coral and the finger corals. And fast forward to 2019, and what we see here is some of the lobe corals remain, but most of this finger coral is gone. So what did we do to gather this information? We use CoralNet, which is an online open source program that uses machine learning algorithms to, once you train it, to then look at the benthos and pick what is there out of it without a human having to do it all the time. So what we did is we did a power analysis, and we determined we needed 40 frames per 100 meter transect to accurately determine percent cover. And then per those 40 frames, we had the computer randomly pick 10 points within it on the bottom third of the view. And then CoralNet would then say, oh, okay, well, this is a rock, a fish, water, or it's some type of coral. And once we trained it, it was actually accurate up to 98%. So what did we find over time? And this is the lobe coral, or Parietes lobata. You can see here, again, this is the marine heat wave line. In Puako, after the marine heat wave, there was a decrease in live corals, which is the lighter line, and an increase in dead, which is the darker line. And this was basically the case throughout, except for this site 5 was pretty stable over time. And then site 3 at Kona, it was the same story, and the same at Mahukona as well. So all of the sites, except for really site 5, which is end of the road Poniao, the lobe coral decreased after the marine heat wave. And then for the finger coral, which was less common at all the sites, it was even more dramatic. So we see here, actually, this finger coral was starting to decrease before, this is live percent cover before the marine heat wave, and the dead coral was starting to increase. So we have to figure out what was going on actually before this marine heat wave. Looks like the finger coral, the Parietes compressa, was getting stressed actually before that occurred. And the same here at Mahukona site 6. These other sites just didn't have that much of this finger coral to begin with. So in conclusion, these unique survey methods allowed us to extract multiple data sets, the fish and the coral. And we figured out that we didn't have to do multiple replicates to accurately assess community structure, which is great. So this last time that we went, we actually added some more sites to gather more information. And then we saved this archive video for future potential projects. Like if somebody wants to look at crown of thorns, stars or something like that, we have that data archived. And the fish communities changed significantly after the marine heat wave events, which increases primarily in the grazers on algae and the planktivores. And then live coral coverage of the two most common coral species were found to decline after the marine heat waves, and actually even before in the finger coral. So what else does the Seattle Aquarium do besides this monitoring and educating our guests about the Hawaii fish and the ecosystem? For management purposes, we share our data with Hawaii Division of Aquatic Resources, University of Hawaii Hilo, and others. We promote certain bills, like we are supporting the Restoring Resilient Reefs Acts. In 2019, we also support the 30 by 30 Now America Beautiful initiative to conserve, connect, and restore 30% of lands and waters by 2030. These are the two papers that have come out of this data set. These are all open access papers. The percent coral cover paper is to be submitted and published early this year. All of our data is open source, so you can access all of the raw data, and then the R code on github.com. If you just search for Seattle Aquarium Hawaii coral reef monitoring, you can see the data. And then what I wanted to share with you, hold on, was just this last month, Site 2 in Pu'uko. Even though the coral looks horrible, it's just amazing to me how much fish there are there. So we really need to dig into the water quality, the food availability for these animals, and other metrics to try and figure out why the fish communities seem to be doing well when the coral really is not. Anyway, that's all I have. Thank you. Should I stop share?
Sure. Shawn, thanks so much for presenting that data and your surveys. Does anybody have any questions?
Let's see, we had one more person join.
Hi, Art.
Hi, hey, Lauren and Shawn. Hi, Kambu and Jay. Thank you for an interesting press. Sorry for my quality of my picture. I just was wondering, is there any kind of, we supposed to measure temperature, water temperature?
Yeah.
And is there a correlation between that and, of course there is, but, something that we can scientifically measure?
Yes, so Amy in her paper did look at sea surface temperature and documented that marine heat wave. And what we need to do, so it didn't negatively impact the fish communities, as you could see, but what we haven't done is to do some modeling and look at that impact on the coral.
Mm-hmm.
I feel that, I mean, obviously the corals were stressed and they seem to be recovering better in Old Kona Airport and Mahukona relative to Pu'uko. Of course, we're just monitoring in one, 100-meter strip transect. When you go deeper in Pu'uko, the corals start to look better. So it's really that more 25 to 30 foot area right where the shelf first drops off from really shallow to about 25-30 feet. That's where we're monitoring. And I know there's some water quality in that area, and I want to get into the weeds with the water quality piece to try and figure out if there's a lot of I imagine there's a lot of nutrients going into that system to then feed the algae and the detritus eaters. And that's probably smothering the coral as well as they keep on getting hit by this warmer water. So I think it's multiple stressors, and I just really need to dig into the other data sets to overlay to try and figure out really what's happening.
Well, we're all pretty much aware of the issues at Puako. I have another question. Are currents a factor?
I think that the currents wouldn't be that much of a factor. I think it's more of surge. So it's going to be the surge. So I wonder if we could get, I don't know if there's a data set of surge for these areas, but I just being there, I know that Old Kona feels very surgy when we're there. And then Site 5 in Puako, which is end of the road Poniao, is right next to a surf break. So there is surge there, and those areas tend to be more stable over time, right? So maybe it is that the surge and the flushing of the water that is keeping the corals.
Excuse me. The reason I'm asking is because Kailua means two currents, basically.
Yes.
And so I was just wondering if the currents had, and the fact that there are two currents in that area,
right,
if that was a factor in it doing better, than Puako or Mahuka.
Yep. I should look into that. If there's a current, there has to be data sets for the currents. Yeah. Yeah, it is confounding that the coral is just not doing well, but then the fish seem to be doing okay. So there's a lot of variables that need to be looked into, and sea surface temperature, currents, and then also its general water quality.
Aside from the bleaching of the coral, do you study basically coral blooms and
recruitment?
Yeah, and then what happens after they bloom?
Yeah, we don't do
that kind of thing.
So we're non-invasive monitoring, taking pictures essentially, and I feel like in order for us to get a handle on that, we would have to put some settlement plates out and to see, or settlement traps to see the coral larval recruitment or spawning events. And I think DAR does that in some of these areas. Yeah, we certainly aren't not gathering all the information that we need to fully understand these systems, but it's interesting that I was encouraged that by taking this video that we could actually see changes in fish assemblages, and that we could see changes in the coral as well.
Thank you, Shawn.
Thank you.
Appreciate it.
And then there was a question in the chat about, have you or others considered combining effects from higher temperatures with other issues from land-based pollutions? You've kind of answered that.
Yes, that's what.
But if you have any more specific details,
that's our intent, is to work with others who have other data sets and then there's publicly available data sets from NOAA satellites, for example, for sea surface temperatures and other things, even dissolved oxygen, even chlorophyll, that we can look at and overlay all of those different variables to try and understand what's happening.
Shawn, when you're talking about, first of all, great job. Thank you for presenting this. I'm glad I was able to attend. I'm new here, so Arik, you said people know what's going on in Puako, and I don't know what's going on. I'm trying to educate myself. You were in here, I worked with Shawn quite a bit in the Pacific Northwest, mostly on some marine mammal issues, but mostly on stormwater runoff, what have you. And as a toxicologist, I want to jump to water quality and say, of course, I wrote in my notes as you were talking multi-stressors before you said it in your answer, multi-stressors. I'd always love to think it's a water quality issue as a toxicologist, but it's always a myriad of things. But when you say they have water quality data, what type of parameters they're measuring?
They're measuring a whole suite of nutrients. So many different types of phosphates and nitrates, as well as fecal coliform bacteria. And basically what's happening in Pu'uko, and Eric and others, and Lauren probably know this story better than I do, but they don't have, they're all on septics or cesspools on lava, right? And there is not a central water treatment plant for them.
Right, that's what I was going to get at. I gave about 3 or 4 months ago, I gave a presentation to UH Hilo folks about the stormwater stuff and 6 ppd and tire dust and what I've learned, basically Eric and Lauren, our research group spent 20 years trying to find out what was killing salmon in the Pacific Northwest after spending billions of dollars to recover salmon, like we are with our coral reefs and what have you, and temperatures involved and many other things are involved. That we came up with basically an antioxidant that's put in your tires that's causing the problem after a 20-year journey. And so, when I got here, of course, I'm in Pahoa as well. And, I think I'm on cesspool, I'm not even on septic. And so they're still measuring nitrogen, phosphorus, E. coli, let alone 6 ppd and PFAS and all those other chemicals that, that we look for in the Pacific Northwest quite a bit because we of all the industry there and what have you. So I guess, again, not knowing the area very well on that side, on the Kona side, what about when you said, Kona, the old Kona site seems to be very stable. Well, I'm assuming just from my time over on the Kona side that, that probably that area off where you're studying has probably been developed and there's not much more development able to be going on there, right? But in the other 2 areas, Pu'uko and Mahu Kona, maybe there's more development at this point going on, and i.e. then anytime you develop something and pave more things over, you have more runoff. And even though you're just measuring nutrients and E. coli, there's probably a whole, we got a lot of tires in here as well. Yeah. There's probably a whole host of chemicals that are coming off. And then the last thing I'll let you comment was that you said, we're measuring there in the zone before it gets deeper. Well, of course, that's where any runoff would be more impactful, right? Number one, Eric talked about the two currents. And then the other time you're talking about doing the monitoring in the shallow. It's probably before you get a lot of mixing of whatever's coming off the land.
Yeah. So Tracy Wigner with University of Hawaii Hilo has tons of data on specifically Pu'uko and relatively shallow. So then we were trying to get some samples where we were actually surveying. So we brought down liter bottles and collected samples, at depth 25, 30 feet down and gave them to her lab to run. And then we did some fecal coliform, we did some legacy chemicals and things like that. And what we found is that one sample at that time just wasn't enough to really understand what was going on. They find a lot more pollution in Pu'uko really shallow than we were finding, or at least fecal coliforms than we were finding at depth. But it's clear that there's, to your point, yes, Pu'uko is the most developed area of all of the sites that we're surveying. So Mahu Kona is a state park and doesn't have any more development, but yet people are driving there all the time with their cars. And so, but, they're trying to sign me out. Anyway, people are driving there and all the time. And then Old Kona is actually also a park, and there isn't that much residential right there. So it used to be the Old Kona Airport. But people again are driving there, and so I agree with everything you said, Jay. I just don't have all the data.
Yeah, no, I'm sure sooner or later, if not Noah and Nat and those folks are going to start looking at coral sensitivity to things like 6 ppd, and that's the chemical that comes off the tires, or we actually put in tires and it turns into a quinone that's very, very toxic to some critters, and then very, very resistant to other critters. So I bet you Noah, before too long, Noah will be doing some of those. Everyone's jumping on it. There's some Canadian scientists and some folks over in Australia now looking at the sensitivity. And of course, the Australian people would be very interested with the Great Coral Reef. And and up and down that Gold Coast is quite a bit of development. So anyhow, people are starting to jump on the bandwagon for some of these new chemicals to see the sensitivity to things like coral and other sensitive critters.
Yeah. And then, so by far Pu'uko's the most residential of these, the areas that we're surveying, but also Pu'uko that used to be smaller homes that have been bought up, and they're replacing these small homes with huge homes that have a more, probably a more impact on the reef, you know, just because instead of being 1 or 2 bedroom, they're building, you know, whatever, 5, 6 bedroom, you know, more luxurious big homes. And then also there are pesticides used to control for the pests, and then herbicides used to control. So, you know, there's Roundup that's being sprayed, and then other things. And we tested for all that again, but found that sampling once a year really didn't give us the information that we needed. We needed more regular sampling. So
Thank you.